Bismillahirrahmanirrahim, and welcome back to this next video. And in this particular video, we are going to talk about the complement system, which is also known as the complement cascade. Now, this is an overall view of the complement system, and you can see the different components of the complement pathway in this particular image. Like you can see the C1 complex, you can see the C3 convertase, you can see the C3b, the C6, the C7, which are the different components of the complement system. Don't worry about them. We are going to discuss them in detail, that how these components they combine with each other to give you a complement system. Now this complement system, like there are different types of the pathways of the complement system. The one we are focusing on in this particular video, that is known as the classical pathway, and we will talk about the other pathways, the alternative pathway and the lectin pathway, in the videos to come. Now, before going into the pathway, the nomenclature of the complement protein, that is very important to understand. So, these complement proteins, they are designated by the capital letter C, which is followed by a number from 1 to 9. For example, the C1, the C2, the C3 till C9. And these numbers, the 1, 2, and 3, they are actually showing the order of their discovery. So the C1, that would be the first complement protein to be discovered, followed by C2, then C3, and so on. These complement proteins, they circulate in the serum in their inactive form, and when they are needed, you are going to get them activated. This would be very much similar to the production of the trypsin enzyme from its inactive precursor, the trypsinogen. So the trypsin is produced in its inactive form, which is the trypsinogen, and when you need the activity of the trypsin, you are going to convert the trypsinogen into the trypsin. Same is true for these complement proteins. You are going to produce them in their inactive form, and when they are needed, you are going to get them activated. Now, how they are activated? So these complement proteins, they are actually activated by cleavage, and when the complement proteins they are cleaved, the resulting peptide fragments, they are denoted by adding a lower suffix a and b. Now, this suffix A and B, in most of the cases, the smaller fragment that results from the cleavage of the complement protein, that is designated by the small a, and the larger fragment that is designated by the small b. Now, C2 is an exception where the things that are the other way around. The smaller fragment in the C2 that is designated by B, and the larger fragment that is designated by the small a. When we talk about this complement system, that is an important part of the innate immunity, and that is also an important part of the humoral immunity. Why we call this as the complement system? Because it complements or it enhances the ability of the antibodies and the phagocytic cells. So as it is complementing the immune system, it is enhancing the immune system. We call this as the complement system. Now, this complement system, it is very important to clear the microbes and the damaged cells from an organism. And this complement system, it consists of a number of small, inactive, liver synthesized protein precursors circulating in the blood. So when you talk about these complement proteins, most of them they are synthesized by the liver, but there are complement proteins which are synthesized by the macrophages as well, but most of them they are synthesized by the liver in their inactive form. About 50 proteins and protein fragments, including the plasma proteins and the cell membrane receptors, that constitute the complement system. There are three biochemical pathways that activate the complement system, as I've told you. The first one, that is known as the classical complement pathway, and this would be the pathway that we'll be focusing on in this particular video. The other one is known as the alternative complement pathway, and the third pathway is known as the lectin pathway. Now, how the classical complement pathway works? Let us focus on this. When there is an antigen in the body, so the antibodies they are going to attach to these antigens. So if this is the antigen over here, this would be the epitopes over here, and this one is the antibody. So when the antibody that combines with the antigen, So you are going to have this antigen antibody complex, and this particular antigen antibody complex that is working as a signal for the activation of the classical complement pathway. And the specific types of the antibodies that activate the complement pathway, that are the IgM or IgG. What I mean to say is that when the IgM or the IgG antibodies that attach to the antigen, so this complex of the antigen with the IgM or with the IgG, that is acting as a signal for the activation of the classical complement pathway. When we talk about the classical complement pathway, you need to understand the C1 complex, because this is the first of the complexes which actually interacts with the complex of the antigen and the antibody. And the inactive C1 complex, also known as the complement component 1, this is a protein complex, and it contains three types of the proteins. One is known as the C1q, the other one is the C1r, and the third one is the C1s. And in this inactive complex, there are six molecules of the C1q, two molecules of the C1r, and two molecules of the C1s. And this whole complex is actually stabilized by the calcium. When you talk about the antibodies, as I've told you, that the classical complement pathway is activated by the complex of the antigen and the antibody. So, how this whole process it works. Now, this is the structure of the antibody. I'm not interested to tell you about the structure of the antibody in this particular video, because we are going to discuss that in detail when we talk about the B cells and then the antibodies. But just to give you an understanding of the classical complement pathway, you need to understand that there is a particular site in the antibody which is known as the Fc fragment. This actually provides a binding site to the C1 complex, specifically the C1q component of the C1 complex. So when the antibody, the IgM or the IgG, that binds to an antigen, that provide a binding site to the C1q. The C1q, it actually binds to the Fc fragment, because the specialized CH2 domain of the Fc fragment, that is providing a binding site to the C1q in the C1 complex. Now, the C1q is actually a recognition subunit. What I mean to say is it is responsible for the recognition of the antibody, and the other two components, the C1r and the C1s, they are the proteolytic subunit of the C1 complex. So, when there is antigen and antibody interaction, the C1 complex that is attaching to the antigen antibody complex, and when this and within this C1 complex, the C1q that is attaching to the Fc region of the antibody, as you can see in this particular image. Now, with the C1q, it attaches to the Fc region. There are conformational changes taking place in the C1q, and that is very much clear in this particular image. This is the inactive C1q, and you can see the conformational change over here. This is the active C1q, and this happens when the C1q that interacts with the antigen antibody complex. Now, in this particular C1 complex, as I've told you, the other two components, one is the C1r, which is actually a serine protease that is also there. So when the C1q, because of the conformational changes, gets activated, it is responsible for the conformational changes in the C1r and the activation of the auto cleavage property of the serine protease C1r. So the C1r, when the conformational changes they take place, it gets activated, and it is going for the auto cleavage, and when it's auto cleave itself, you are going to get the active C1r. So you can actually say that this conformational change is actually responsible for the activation of the serine protease property of the C1r, and because of the activation of the serine protease activity, it is auto cleaving itself, thereby converting itself into the active form. So now you have got the active C1r. So when the C1r it gets activated, what it do is it is going to act on the C1s, and when it acts on the C1s, you are going for the cleavage of the C1s by the active C1r, and that is going to give you the active C1s. So this is how you are going to activate the C1s. When we talk about this C1s, it is responsible for the cleavage of the complement proteins. So the active C1s, it is responsible for the activation of the C4 complement protein as well as the C2 complement protein, as you will see in a while. So when the activated C1s, it acts on the C4 complement protein, it is going to cleave the C4 into two components. One, the C4a, and the other component, the C4b. So the activated C1s is responsible for the cleavage of the complement protein, and I've told you when they are cleaved, you are going to get the active components. So the C4, when it is cleaved into the C4a and the C4b, you are going to get the C4 activated. So this is the C4b, because when you talk about the complement pathway, this is usually the C4b, which is responsible for the formation of a complex on the target membrane, as you will see in a while. The smaller fragments that are usually responsible for mediating a local inflammatory response, but they are not responsible for making the complex, the MAC complex, that we will see at the end of this particular lecture. Now, when this C4 it gets activated, the C4, especially the C4b, so this C4b that is attached to the surface of the you can say pathogen surface. So if this is the surface of the pathogen, the C4b that is going to bind covalently to the pathogen surface. Now, when it binds to here, the other component in the pathway that is activated by the C1s, that is the C2. So now you are going to activate the C2, and again, this activation would be carried out by the C1s. Now, if you look at this one, this C4b, this C4b is actually providing as a receptor site for the binding of the C2. So you can say that this C4b is actually a receptor, or it provides a binding site to the C2. So when the C2 that binds to the C4b, this is actually the binding of the C2 and the C4b that results in the C2 being cleaved by the activated C1s into the C2a and the C2b. So when the C2 that attached to the C4, the activated C1s that would act on the C2, thereby giving you again two fragments, the C2a and the C2b. Now, when the C2b, in this particular image, when you get this C2b, you are going to talk about the other complex, which is known as the C3 convertase. So how this C3 convertase that is formed, and what are the functions of the C3 convertase, we will talk about that in the next video.